Inside almost every cell is an instruction manual. It contains 3 billion letters, documenting the exact specifications for every biological function keeping you alive. Looking at this physical DNA structure, you see how precise these instructions must be. A single incorrect letter disrupts the system, causing lifelong inherited diseases. For decades, if you inherited a faulty sequence, doctors could only treat the downstream effects. They managed the symptoms because the source code was entirely out of reach. Gene therapy acts as a molecular find and replace function. It locates the specific typo and swaps it out for a healthy, functional sequence. Once that healthy sequence successfully integrates, it delivers functional code directly into the cells to compensate for the faulty one. By fixing the mutation at its source, we move from a lifetime as managing symptoms to a one-time permanent correction. But getting that new sequence into the body poses a massive biological challenge. If you put free-floating DNA into the bloodstream, it simply bounces off the impenetrable exterior walls of human cells. To get inside, scientists need a specialized vehicle. They use what are called vectors, specifically engineered viruses that have been completely hollowed out and stripped of their disease-causing elements. As seen in this diagram, the viral vector, shown as the pink shape, attaches to the membrane and sneaks its payload inside. From there, the therapeutic DNA travels directly into the nucleus, integrating into the host's genetic structure. There are two distinct methods. In ex vivo therapy, doctors remove target cells, modify them with the vector in a lab, and reinfuse them. In vivo therapy, the vector is injected straight into the patient, bypassing the lab entirely. On paper, the mechanics of this delivery system worked. But moving from theory to a living human system meant introducing modified viruses in doses that carried an invisible and catastrophic risk. In the late 1990s, immense scientific optimism fueled the field. Labs like this one rapidly moved early gene therapy concepts into aggressive human trials. That momentum collided with reality in 1999. An 18-year-old named Jesse Gelsinger volunteered for an in vivo trial to treat a rare metabolic liver disorder. When researchers injected a massive dose of viral vectors, Jesse's immune system didn't recognize them as a cure. It identified the dose as a lethal invading threat and immediately mounted a severe attack. This immune response triggered uncontrollable inflammation throughout his body. White blood cells overwhelmed his organs, and within days, the reaction proved fatal. Jesse's death brought global gene therapy research to an immediate halt. It proved that theoretical genetic cures were utterly useless if the delivery vehicle itself killed the patient. The scientific community spent the next decade entirely rethinking cellular delivery. They redesigned viral vectors to be highly stealthy, allowing them to quietly evade immune detection. They also developed entirely non-viral delivery systems. One major breakthrough involves placing the fragile genetic payload inside a microscopic fat bubble, known as a lipid nanoparticle, so it can slip harmlessly into the cell. This diagram illustrates a surgical approach, CRISPR. Instead of replacing an entire gene, it allows the direct replacement of specific mutated sequences within the double helix. Guided by RNA, it zeroes in on the exact mutation and repairs it directly within the DNA strand. We have now moved far past the high-risk experiments of the 1990s. Today, in highly regulated clinical environments like this one, gene therapies are actively deployed in standard patient care. People born with previously incurable rare genetic diseases are now receiving single-dose treatments that permanently rewrite their cellular code. After decades of early trials, through tragic clinical failures and relentless technological iteration, medicine finally has the ability to safely rewrite the basic text of human life.